Hi, good morning. So today I'll be talking about our effort to atlas human liver during a disease, mainly cancer. So this is a work in collaboration between Ram's lab at GIS, Professor PS at NCCS, and Floron at SSGN. So since last 3-4 years, I'm interested in looking at tumor evolution and how tumor evolution follows some of the developmental pathways, right? So we were looking at how tumor evolved under selection pressure of the chemotherapy, and this we are doing in context of head and neck cancer and breast cancer. But if you think about tumor evolution starts much earlier than that. It starts when a normal cell gets mutated and start for its survival in a niche which is full of immune cells which are ready to kill it, right? So we wanted to interested how a normal cell survive when it gets mutated in a immune system. And we thought we'll work on the liver because this is one of the best organ. It's a highly regenerative organ, and some of the hypothesis of cancer stem cell tells us that cancer is the price of regeneration which we are paying, right? So when we started working on this, So basically what we are trying here is trying to make an atlas of liver from homeostasis to disease, trying to get enough healthy samples and inflamed samples like NASH and HCC. So this work is partially supported by CGI and mainly supported by a TCR grant to Professor PS. So what we do in this part of the TCR program is that we are collecting these liver cancer samples, and we are doing this multisectoral sequencing, and then the sample is sent to the different modalities, if you say so, metabolomics, genomics, transcriptomics, and we are looking into the single cell part. Also, most importantly, these cells are also modeled as a PDX model, so whatever the observation we get from our atlas, we can go back and validate in mouse model. So this is a large collaborative network where we are getting samples from most of the countries in Southeast Asia, including 2 sites in Singapore. And this is a very nice example of a common coordinated framework where we know which site of the tumor we are getting. We divide this tumor into multiple parts, and we do this multisectoral sequencing. So as of now, there are not too many data available on single cell where we know which part, doing multisectoral sequencing where we know how the profile looked at the periphery versus core. So when we started this thing, the first problem we faced is that, which most of the people who are working on tumor single cell may have faced, it's very difficult to get epithelial cells out. And this problem is much bigger in liver because hepatocytes are more fragile and they don't like to be dissociated or fragmented. So we overcome this problem by mixing the CD45 positive and negative cells in 1:1 ratio to get a decent recovery of both immune and non-immune cells, because eventually you want to comprehensively profile all the cell types. Also, as I mentioned, because we are getting sample from multiple sites and sample comes at different time points, so we wanted to optimize the different freezing conditions. So in this case, we optimize how sample will look like at the fresh freezing and in a stimulated situation of dry shipment. So from same sample, we are doing all these 3 profiling. And when we make this, when we profile this, annotate all these cell types, we ask the question, which cell types are most affected by freezing? And if you see, there are 2 cell types which we found were most affected by freezing. And these 2 cell types were the most important cell types which you want to look into liver, hepatocyte and Kupffer cells, right? So if you see that in fresh, you see almost 10 to 15% of hepatocyte and Kupffer cells, but after freezing, you lose these cells. We dig a little bit more deeper into this and why this is happening, and we found that at fresh, when we are looking at the profile of the fresh samples, there's a bimodal distribution of the RNA. But during dry ice and -80, you see this species which is expressing lower number of RNAs completely gone. And same is true when you look at the individual low-in clusters. And the cluster which we specifically lost during freezing were hepatocyte and Kupffer cells, and some of these cells were expressing less RNA. When we looked specifically into hepatocyte, because in hepatocyte we see this bimodal distribution to begin with, interestingly, there's this one cluster. We completely lose this cluster 5, which is in brown here. And you see these samples are only coming from the fresh. And if you look at this cluster does not express too many genes to begin with, but it is highly enriched for albumin and AFP, so suggesting that this freezing might be affecting your stem-like cells. So we were interested in hepatocyte and Kupffer cell. That's why we did not freeze our cells. And at least for liver, we think we have to work with fresh samples all the time. So with this information in mind, we started making this atlas. And in this atlas, we had 14 patients, multi-sector sequencing, so almost 60 sectors, encompassing adjacent normal and tumor, and one healthy normal as of now, but we are trying to increase this number. And we sequence these 74,000 cells approximately and find this 29 mega cluster. When we individually break down these clusters, we see the more number of cells, and I'll give you some of these examples. But since we were looking at the different sectors, we could easily identify this population, which cells are coming from the periphery of the tumor and which cells are coming from the core of the tumor. Because in literature, we know that tumor core is more hypoxic, so there could be different cell composition compared to the periphery of the tumor. So in this UMAP, you see the yellow is from the normal, red is from the core of the tumor, and purple is the periphery. So as you can see, there's a great distribution of or distinction between the cells from different part of the tumor. If you look at the myeloid cluster, one cluster is mainly coming from the core and periphery, while other has some component from the normal. Immune cells are very interesting because you see some immune cells are coming mainly from the normal, while other coming from the core and periphery. And hepatocyte, we see some cluster specifically to the core, which means that core versus periphery may have different blood requirement, and that's why you have different kind of endothelial cells. So we started annotating this cluster, and we found individual marker for each and every cluster. And then we started going deeper and doing the second level of clustering, where we take the lymphoid cells and subcluster it further. So when we subcluster the lymphoid cells, so these are the T cells, and we found there are almost 20 cell types or cell states, how you want to define them. But one of the most interesting things is that we could find two different kind of Treg populations, and you see these Treg populations are coming mainly from the tumor and coming from the different patients, right? So which means we are not finding a population which is represented by just one or two patients. Similarly, we found that when looking at another cytotoxic population, NK cell, they are mainly coming from the normal. Now, if you look at the cytotoxic cells, these are CD8 positive cells here. So these cells are mainly coming from the normal again. So we suggest that liver is an example of cold tumor. We don't see too much infiltration of the cytotoxic T cells. Well, there's a lot of CD4 positive cells, and specifically Treg cells are present in the periphery and core of the tumor. We look at another immune population, which is supposed to play an important role in immune modulation. So that's why we look at the tumor-associated macrophages. And in this cluster, we are looking at clustering all the myeloid cells. And if you look at these cells, these are the TAM populations. So we found three different TAM populations in the liver, TAM1, TAM2, and TAM3, and we found individual marker of these. And if you look into this UMAP, these cells are mainly coming from the core and periphery. We do not see any distinction between TAM1 and TAM2 more or less in periphery versus core. Next, we use these protein markers, and with the help of Charles and Florence lab, we validated this in an independent set of experiments using flow cytometer, and based on CD163206 and panel of another markers, we could identify these TAM1, TAM2, TAM3 in the human patient looking at the normal and tumor, and you see these cells are not present in normal to begin with and highly enriched in the tumor. And this is looking at each and every sector. So these red cells are the TAM1 population. As you see in normal, these are highly or like almost not present, but they are highly enriched in the tumor. And then we did RNA-fish experiment to validate these two cell types, TAM1 and TAM2 marker are coming from the two different populations. So I'm not sure. So if you look at this is the normal CD163, there are very few CD163 cells, something similar to this, but they increase in tumor. Well, the TAM2 marker, SPP1, we do not see in normal, but they are highly, but we see a small population of the SPP1 in the tumor. Next, we wanted to understand if these populations are present, which are specific to the tumor, Tregs, myeloid cells, are they interacting with each other, right? So we use this cell phone DB approach to look into the different receptor ligand interaction. But before that, we just looked at this in a very holistic way. How many interactions are there in normal versus tumor? Which kind of interactions are increasing in normal and tumor? So when we did this analysis, we found that there are twice the number of interaction between macrophage and T cells compared to normal and tumor. And when we specifically look into these interactions, we found these interactions were immunosuppressive in nature. For example, you see there's an increase of CD86 CTLA4 interaction, CD28 CD86, CD58 CD2, and CD47 and SPICBA. So, but this interesting part was that these macrophage interaction, we were seeing both CD4 cells, CD8 cells, and NK cells. But we know that in tumor, CD8 and NK cells are not that much. That's why we specifically looked into the interaction between Tregs and macrophage population. And now we are looking at two different kind of macrophage population. Since there are three macrophage population, we wanted to understand which one is more immunosuppressive, right? So if you look at this profile, this is the TAM1 population, and as you can appreciate, in this dot plot, there are more immunosuppressive interaction where TAM1 is involved compared to TAM2. And these are the marker of the TAM1 and TAM2 population. And TAM1 express more CXCL12, CXCL16, CD86. So suggesting that in this, in HCC, what we are finding is that the different TAM population, among these, TAM1 is more immunosuppressive compared to TAM2 and 3 and involved in interaction with Tregs. And finally, we looked into the RNA fish, and we are looking at the CD163, SPP, and TIGIT here. So as you can see, the CD163 cells are more close to the Treg population compared to the TIGIT population, suggesting that CD163 might be involved in more immunosuppressive interactions. And so this is what we think is happening in HCC based on what our atlasing we are doing right now is that this is an example of a cold tumor. There are less cytotoxic cells, more immunosuppressive population. And what we also found is that these interactions are specific between macrophage and T cells, right? So what we are doing from now, this is that now we have the marker of these macrophage populations. We have marker of some of these T cells. We are using Garnet kind of methods to make a signature of these TAM population and query another data set. Specifically, we are interested in querying our data set because it's a prospective data. So at some point of time, we'll start getting the relapse patient. We are also interested in looking at this profile, how these change. It can tell you about patient who relapse early versus late, and what it can tell us about the immunotherapy responder versus non-responder. So that's another area where we are interested in. Some of the early observations we have, some of these patients are given this treatment Y90, and what we have profiled so far that after Y90 there is a decrease in this population, which make it more, which lead to the increase in CD8 positive cells in the tumor. So that's another part where we are looking into. So to summarize, I have shown you our initial effort of making HCC Atlas, where we have profiled 14 patients, 80,000 cells from 58 sectors, 15 normal, 43 tumor. We found 29 major cell types, but there are many more cell states. It shows the sign of cold tumor, 3 TAM population, one of them is more immunosuppressive. And now we are interested in looking at if this TAM could serve as a biomarker of the drug response and how these interactions, so right now we have just talked about the interaction between T cell and macrophage, but how these interacting with other stromal cells, for example, fibroblast or endothelial cells. So in the end, I would like to thank Ram to supporting me in this work and mentoring me. Justin did most of the bioinformatics work, and Ria and Hermin for help in tissue dissociation. Florence lab, especially Charles, for helping us with teaching all these immune cells, myeloid cells, and DC nomenclature, which I still don't get it. Professor Pierce for helping in this project and mentoring us throughout this process. And as I said, this is supported by NMRC, and this is Justin and Ria. Thank you.
Thank you, Ankur.